Okay, we're set.
Great. Well, good morning, everyone. I am Deborah Atwood, Executive Director of AGREE. And on behalf of AGREE today, I would like to thank you for joining us for the Gene Editing 101 webinar. And I think the information shared today will help us all prepare for the upcoming convening Friday called Emerging Gene Editing Technologies in Agriculture. I'd like to acknowledge the presence on this call of two great thinkers on the subject who will be co-chairing our upcoming meeting. First, Jim Burrell. He's recently retired from Dupont, where he served as Executive Vice President, and Greg Jaffe, who is the Director of the Biotechnology Project at the Center for Science in the Public Interest. I'd also like to introduce John Ehrman, who is the Managing Partner with the Meridian Institute. Meridian serves as AGREE's institutional home, and John is going to serve as our moderator for the webinar. So I'm turning it over to you, John.
Thanks, Deb, and thanks to everyone who's on the line. And for those on Adobe, you can see the various folks who are with us on the line. I think what we'll do is go ahead and get going, and then perhaps when Neal and Jennifer are done with their presentation, we'll just take a quick check to identify everybody who's with us, because I'm sure we may have a few other folks join as we go forward here. Let me just say a word about the purpose of this webinar. As you all know, we're having a dialogue session on Friday on this in our office in BC on this very important topic. And in preparing with the co-chairs, we all felt that it would be helpful to have an opportunity for a bit of a 101 learning session, so that when we all get together on Friday, we can, as quickly as possible, get into the issues associated with these technologies and spend relatively limited amount of time explaining them in terms of what's basically on the table relative to these emerging technologies. So we're very pleased that Neal Gutterson, Vice President of Research and Development at Dupont, and Jennifer Kuzma from Goodnight-NCGSK Foundation and Distinguished Professor in Social Sciences and Co-Director of the Genetic Engineering and Society Center at North Carolina State University have joined us to provide that overview. And we've asked Neal and Jennifer to really keep this to the 101 type approach in terms of describing the range of technologies, et cetera, and we'll reserve the discussion of opportunities and concerns and issues associated for our discussion on Friday. So I'm going to turn it over without any further ado to Neal and Jennifer here in a sec. Neal will go first, and then Jennifer, and then we'll have some time for Q&A and follow-up questions from all of you to our presenters. If you wish to type questions, you can do that if you're on Adobe in the question and answer box, which should be up in the top right-hand corner of your screen. And that will give them a heads up to the kind of questions you have as they go forward. With that, let me turn it over to Neal to get us started.
All right, thanks, John. And it's a pleasure to be here and virtually and take part in this pre-conference webinar or pre-workshop webinar. So I'm just going to jump in. Let's go to the next slide. And I just want to start with, so I'm going to do some grounding here into the focus of the technology, and I think the area that the workshop is going to try to focus on. So this first slide just lays out the fact that when it comes to genome editing, there are a number of tools and technologies that have been developed over time. And in the last few years, the excitement about the potential of genome editing has really soared with the development of the technology on the far right, CRISPR-Cas technology. The slide shows an assessment that DuPont pioneer scientists have done of those different technologies. And you can see in terms of time efficiency, quality, cost efficiency in a dramatic way, technical flexibility, CRISPR-Cas is a very much superior technology, and that's why the level of interest in genome editing has soared, because of the potential to actually implement what's been an interesting concept for quite some time. I want to just say also at the outset that any of these tools can be used in a variety of ways As you'll see in a minute, gene editing is a particular approach to targeted changes in genomes. And amongst those targeted changes would be the ability to deliver a transgene in a targeted way, to create a biotech trait. And in that context, all the issues that we're familiar with today about GMOs are the same. But what's really exciting about this technology from the standpoint of Dupont Pioneer and many, many people in the agricultural community is a different set of applications, which is really where I'm going to focus on for the next 15 to 20 minutes. And that is in a context of advanced plant breeding. So if you go to the next slide, John. So here's how we think about this technology, really the meaning of the technology for agriculture. So CRISPR-Cas is an advanced plant breeding tool, and you can read the words on this slide. I'm not going to read them all to you. But the key is that it's a more efficient and targeted plant breeding technology. And the why of this is the why that all of us in agriculture have cared about for a very long time. We know that farmers face real challenges. The pace of change of those challenges and the stresses that they face, climate change, drought, disease is accelerating. Population's growing. We're familiar with all of these issues. The why then about this technology specifically, in addition to that broad why, it has to do really with the ability to be more efficient and to bring products more quickly to the market than we otherwise can do. And that's not just for a company like Dupont Pioneer, but for small companies, for academics thinking about this, and also for international ag organizations. So all of these groups can participate, we know, with this technology. Ultimately, in the far right, the what in the simple context is, that second sentence really captures it in our view. It's a more targeted way to develop healthy seeds using the best native characteristics available within the crop. So if you go to the next slide, let me just talk then and give you, I'll come back to this and walk you through a bit about what's behind the ability to edit a genome, but here's the essence of it. And I want to bring your attention to the analogy to a word processor. Because when we talk about genome editing, we're talking about editing the genome as a text, much as one would edit a text that you might have on your computer. And the key to that, if you've got a large text, is to search for the location you want to edit and then to actually initiate that edit. CRISPR-Cas is a system that is very much like that. It is a search and replace type functionality. And with that, we can do three types of things. One of those is to delete a word or to delete a gene in the genome. And so I'll come back to explaining a bit more about how this works, but we can cut at a location and actually drop out. If we cut twice around what you can see there as a gene, we can drop out that entire gene in the genome. By analogy, in a text, you might start with a phrase like, the plant does not have desired quality output, delete a word like the not, and end up with the plant having desired quality output. So deleting is a very powerful function of the CRISPR-Cas genome editing technology. We can also edit. And so rather than deleting an entire gene, we can make a very small change. So we know that, for example, the corn genome has in the neighborhood of 3 billion base pairs, very similar to the size of the human genome. We can go with a search and replace function to one very particular place in the genome and modify a single base in the genetic code. And the analogy to editing might be that you have a text, and the text might say the plant has tolerance to drought, and we can edit that to deliver a plant that has high tolerance to drought. So that's the edit function of a genome editing process like CRISPR-Cas. And finally, we can have the idea of a replace function. And so you might want to actually replace a word in your text. You might start with a text like the plant is susceptible to disease, want to edit that and make the plant resistant to disease, replacing that word. In like fashion, we can either swap a gene that gives resistance with one that is sensitive, for example, to a particular disease, or in some cases, one variety might actually not even have that gene. Another variety would have the gene, and we can put that gene from the variety with that gene into the exact same location in the variety without that gene, exactly the same sort of outcomes we would get through breeding. All right, so the next slide then. Just want to step back and put this in a context of the landscape that we see in the greater world today beyond agriculture. CRISPR-Cas is a technology that colleagues at Dupont worked on for many years, earlier in this decade and even before, to understand a system that exists in bacteria to affect resistance to viruses. And so you can see in the earlier, the last 5 years between, say, 2007 and 12, there were a handful of publications. In 2012, the first publication was made, Jennifer Dowden at UC Berkeley, on the particular application of CRISPR-Cas to genome editing, and you can see an explosion in academic publications that appeared from 2012 on to 2015 and continues to grow. And so the academic community, the scientific community, is extremely interested in this technology because of the many applications. At the same time, we can see that there's also tremendous interest in beyond the academic community in this technology because of the potential uses of this technology, not just in agriculture, but in engineering any organism.
Neal, this is John, let me just interrupt for a second. Folks, if you could go on mute if you're, while we're listening to Neal and Jennifer, that would be great. So use your mute function if you can. Thanks.
Right, thank you. So again, we're on the next slide, I think, slide 6. So I'm having trouble with my internet connection here, but if we're on slide 6, what we know is that in 2015, Science Magazine dubbed CRISPR-Cas as the scientific breakthrough of the year, and that's a reflection of the many uses. And you can see, major media like the Los Angeles Times, the New York Times, The Economist, more specific journals like Wired have covered this technology. And the potential therapeutic applications are very substantial, for example. The ability to, for example, take someone's blood cells that might have a blood disorder and modify those cells and put them back into that person directly. The potential therapeutic applications to cure diseases are enormous. And so a lot of excitement, and you can see a lot of concern and anxiety as well. There is potential to engineer humans, the potential to engineer in a way that's heritable, and that has raised substantial ethical concerns that many in the community are wrestling and working with. So let's now turn from that context to the next slide. And I want to anchor the discussion then about the technology into a lens from Dupont Pioneer. How do we think about breeding and what we've done over the last 90 years in our company since its inception in breeding? And you can go back to the picture on the lower left. Copper Cross was the first hybrid developed by Henry A. Wallace, our founder. And that was the beginning of a revolution in plant breeding based on hybridization, shifting from open pollinated varieties of corn where the rate of gain for farmers was very, very limited, to one in which hybrid systems have enabled us and history of plant breeding and germplasm improvement to continuously improve the varieties that we deliver and the yields that our farmers get, and therefore their profitability. And so on the bottom you can see in the series of varieties that we've brought out over the past 90 years, you can also then see the next major revolution in crop improvement, agricultural biotechnology. We participated in that. We have products in the market that are, like Plenish High Lake soybeans and products related to insect control. And agricultural biotechnology is a technology that's going to be around for a very long time, as is hybrid systems and plant breeding as a core. And then building on top of that, we now see CRISPR-Cas advanced breeding as what we believe, looking back in 20 years, we'll see as potentially as revolutionary and as impactful on bringing better products to the market as these other technologies like ag biotech and hybrid systems have been. And at the bottom there it shows our first product that we are looking at bringing out, which I'll come back and mention again a little later. The next slide then, let's anchor this. Now talk about the technology, and I want to anchor that into natural processes that occur and the dynamic nature of a genome. So I mentioned the corn genome is 3 billion bases, the wheat genome is 15 billion bases. These are, or 17 billion bases, very large text, very large genomes. And all the time they're subject to stresses, whether that's heat stress or stresses from radiation or oxidative stress. And as a result of those stresses, the double helix is broken at a single place but in both strands, a double-stranded DNA break. You might imagine that that break, if it's not repaired, is going to be very deleterious to that organism. And so all organisms have evolved mechanisms of repairing those double-stranded breaks. And those repairs can either be highly accurate, in which case the final DNA sequence is exactly the same as the one prior to the break, but they can also be a little bit inaccurate. And we end up with small changes in the genome, and that process is what we then capitalize on with a system like CRISPR-Cas to actually deliver targeted changes to the genome. The two important points here: these are naturally occurring processes, and the genome is highly dynamic in any organism, and certainly in all the crops that we work with and others work with. So the next slide then puts that in context and shows how we use CRISPR-Cas, this search and replace function, to capitalize on these natural processes, based on double-strand breaks. So, two components of CRISPR-Cas: the Cas9 nuclease. A nuclease is an enzyme that cuts nucleic acid, in this case DNA, and it cuts it in two places. You can see the two scissors in the picture. And so it's a cutting function, is the Cas9 nuclease. And the key piece then is how do we guide that nuclease function, that cutting function, to the right place in the genome where we want to make a targeted change. And the way we do that is that protein, that nuclease, actually is associated with what we call the guide RNA, and the guide RNA has a roughly 20 base pair region that is identical to a particular location in the genome, and that actually then brings that nuclease function to create the double-stranded DNA at a location that we have selected. And so once that happens, then a series of processes can ensue that can lead to either that delete outcome, the edit outcome, or the replace outcome. And the next slide then just comes back to that same picture I started with to anchor back to the processes that we want to talk about in terms of genome editing. So if you go to the next slide, again, it shows that picture of deleting, editing, replacing in the context of a text, and bearing in mind that, again, a genome is just a very large text. A book might be maybe a half a million characters, and a genome could be a half a billion or more characters, for example. So very large text to be edited. And again, those same steps of deleting, editing, or replacing that can be done. That's the domain of advanced breeding type tools that we want to focus on, I think, for what's new about this technology from an outcome standpoint. So the next slide, what I want to do then is just now to talk about, for a few slides, how this is a technology that is a step change in breeding, and put that in the context of what breeders do and breeding outcomes. So first of all, you can see in their slide that you can start with a single plant, like the wild mustard plant on the far left, and through simple breeding and the variants that occur in nature, deliver a wide range of outcomes. So breeding can be used to deliver product diversity. If you go to the next slide, you can see now that the next slide, the technology is the key to improving breeding processes. And so Pioneer has been developing for the last 90 years technologies that enable us to increase the rate of gain. And I think you can see in that slide is an arrow. Those red dots sort of align with that arrow, and this shows corn genetic gain increasing over time, and a series of technologies, whether they're using computers to predict outcomes of new varieties, whether they're using wind machines to test the ability of our varieties to withstand wind damage, now to CRISPR-Cas as a new tool. It's now this CRISPR-Cas is part of an evolution of new and improved ways to breed. So that's the broad context. Let's now talk about the next slide. One of the main ways we envision using this technology, and others do as well. So what does a conventional breeder do? what's the challenge that they're faced with? So you might have, for example, on the lower left, a very high quality plant. It's an elite variety. It's adapted to a particular location. Let's say it's adapted to Illinois, but it's sensitive to drought. On the other hand, we have a variety tolerant to drought, but it's very poorly adapted to Illinois. And what you want to do is take just the gene or genes that give tolerance to drought and move them into that elite variety that's well adapted. Conventionally, that's done by a series of crosses where one gets, gets to a seed and regrows a plant and gets another seed and regrows a plant. And in each stage, you can increase the amount of the genetics that comes from the elite variety and limit to just the genes that came from the variety that has the tolerance that we're interested in. And this might take 8 or 9 or 10 generations to get back to a variety that has the traits we want in an elite background. And if there are 2 or 3 traits to do this with, it's a very, very cumbersome and potentially long-term process. So next slide then just shows that using a tool like CRISPR-Cas, where we can make a directed change, a targeted change for that desired characteristic directly in an elite variety, we can do these kinds of changes in only 1 or 2 cycles versus the many, many cycles it would take in a conventional breeding process, even with the most modern methods. And so you can imagine then that the speed with which we can bring a product to market is substantially increased, but it's all in the context of developing that new variety, not shortcutting any of the steps we would go through to test the variety and its performance and fitness for the market. So the next slide then, what I want to do is just make a quick point that this technology has come along at a time when other core competencies have also evolved to enable us to use this technology really well. And so understanding the elite varieties we want to work with, knowing the genome sequences of those varieties, being able to deliver the CRISPR-Cas system directly into those elite varieties, as well as the overall technology itself. All of these tools together are important tools that, although this technology you may have heard is easy, you can do it in your garage, it's not so simple if you want to engineer a new variety. And it takes a lot of capabilities to do that. And because of that, we're actually Pioneer, working with other groups to help them implement the technology, and I'll come back to that in just a moment. So next slide just shows the range of opportunities, the ways we can use the technology. There's a piece missing on the left slide there, but it talks about yield and disease resistance. It talks about increased nutritional value, quality of a soybean oil, for example, or protein. There's a wide range of characteristics that we will apply this technology to, and others will apply this technology to. And you can apply it to a wide range of crops, which we're doing already. And we know it'll be applied to a much wider range of crops than we can work in ourselves. So I just wanted to capture that sense of a very broad application set. It's not simple to use a technology, but it is a very, we think will be a very democratically used technology by many people across the agricultural landscape. So let me end with just a couple of quick slides. Next slide, that shows how we are starting to use the technology. This slide shows the first product that we will bring to the market as Dupont Pioneer. It's called waxy corn. It is a variety that delivers a specialty starch, and I won't get into the characteristics of it, but it's used in food and non-food uses. It's identity preserved in the marketplace. It's a very familiar trait. Wax has been around for a very long time, and the biology is very well known, so therefore we could make these changes in a very straightforward fashion. We went through a process started late last year of submitting a letter to the USDA. It's what's called an Am I Regulated letter to ask whether they would regulate this waxy corn, new and improved waxy corn variety under the BRS regulations. And we received a note back in April telling us that this would not be regulated under those regulations and would be treated as a conventional variety. So that's just the beginning of a longer process of working through U.S. and global regulatory paradigms to be able to bring this product to the market. The next slide just gives a very quick perspective on the product itself. We sell waxy corn today. We're a major provider of waxy corn varieties. but the varieties we develop have some yield disadvantages compared to other varieties we can bring to the market, and that has to do with that process of sequential crosses to create the waxy corn varieties. And so what we've done is to actually be able to delete that entire, what we call the waxy gene, and develop a new and improved variety that has all the performance characteristics of our most elite hybrids with that waxy characteristic at the same time. And so the market will see this in very much the same way, but our farmers will actually have a better product to sell. And finally, one last example. I mentioned that we are working with others and trying to enable other groups. We have a very collaborative model with this technology, recognizing that value is well beyond what we can derive ourselves. This shows an example of a public-private partnership we just announced about 2 weeks ago with CIMMYT, and it's focused on delivering more rapidly to smallholder growers in Africa, initially, a MLN, maize leaf necrosis disease-resistant hybrids of corn. And you can see here that MLN is a really serious disease in Africa, and CIMMYT felt this was a really important problem to solve and believed that this technology can really accelerate their ability to bring new varieties out. And we think this is a great example of the kinds of collaborations that I think we're going to see much more of in the future. And I'll just end with the comment that this is World Food Prize Week in Des Moines. We have lots of folks coming in from around the world and lots of conversations set up with groups like CIMMYT and others to talk about the kinds of products and traits they want to develop for their customer segments. So I'll stop there and hand it back to you, John.
Thank you, Neal, very much. Very, very helpful. I think I'm going to suggest, Jennifer, that we go right to your presentation, and then we'll open it up for questions of clarification or requests for more information.
Great. So I also appreciate being here and being a part of the meeting on Friday, and I'm looking forward to the conversation. I was asked to talk about more the policy context for gene editing today, and so that's what I'm focusing on in the interest of not solving any of the issues, but providing some fodder for discussion on Friday and taking more of the public standpoint. Next slide, please. So as you know, we are in the second generation of genetic engineering and possibly even approaching the third, where we have many different types of products, and gene editing is playing a huge role in this, in both the plant-based genetic modification world as well as the animal. So I will talk a little bit about the animal, agricultural animals and livestock world as well. But this second generation is going to be a really interesting one as gene drives and genetic engineering in the wild comes into play, as well as more and more animals come into the market. Next slide, please. So we have an opportunity, I think, here to re-envision how we govern genetically engineered or genetically modified or bred, whatever terminology you want to use for gene editing, those kinds of products. So I want to talk a little bit. I've been working on gene editing for about 7-8 years or so, which is kind of unusual in that we were looking at this issue pretty much before it was even called gene editing. So next slide, please. So back in 2011, we published an article that questioned whether or not gene-edited products would come under the jurisdiction of the coordinated framework for the regulation of biotechnology. And not to get into too much detail on that framework, because I'm assuming that many of you are familiar with it, but the idea there was that it's the product that should be the focus of regulation, not the process, and that we didn't need any new rules. And so we could use existing rules in order to regulate the products of biotechnology, and this was developed back in 1986. And so there's a couple definitions under the coordinated framework that gene editing challenges. One would be the definition of recombinant DNA, or what is genetic engineering. And then the second would be the specific definition of the regulatory agencies under their various statutes. And for USDA, that would be the Plant Protection Act right now, or the PPA, which dates back to the Federal Plant Pest Act. And so we, in this article, questioned, was trying to raise awareness about whether or not what we called at the time targeted gene modification, or gene editing, would fall under the regulatory system or fall outside these regulatory triggers. And I think today we haven't, there's still quite a bit of uncertainty about that, maybe not so much with USDA and point mutations using gene editing, but with some other changes using gene editing, and there's still quite a bit of uncertainty with how we're going to deal with gene editing in animals and animal products. Next slide, please. So one other thing, we worked with Dan Voitis on this. He was one of the developers of zinc finger nucleases and talons, and we did surveys and interviews with experts to see how they envisioned how these products should be treated in the regulatory system. And we found a diversity of views, you know, from let's treat them like conventionally bred plants, let's treat them like we did genetically engineered plants, and then, you know, some other views somewhere in the middle saying let's strengthen the system just in general because it has a low capacity and there are going to be a higher volume of products coming into the marketplace with gene editing. It's, as Neal pointed out, it's a bit easier to do, and we can have a huge number of products that the regulatory agencies are faced with overseeing, and that could overwhelm the system. So the attitudes were diverse, and they weren't always dependent on the view of the technology or the view of the risks. Next slide, please. We also did a little bit on the mapping of the research in this area, and you know, at the time we stopped at 2011, but the USA was a big leader as far as targeted genetic modification or genome editing, and it was somewhat concentrated, and we found we included both biomedical and agricultural, and of course there was a huge focus on biomedical. We looked at different partnerships between developing countries and found that there were a few with the U.S. and different focal points in the kind of science that developed versus lesser developed countries were doing. Also, we found that there wasn't very much collaboration among funders in this area to fund the work. So that's sort of trying to get a sense of the landscape at the time. Next slide, please. And then I've also done quite a bit of work over the past 15 years on looking at the coordinated framework and trying to evaluate it from more of a policy sciences perspective using multiple types of criteria. And I won't talk about that, but just suffice it to say that what I'll say next is informed by a lot of that work. Next slide, please. So I wanted to just throw out some points for discussion for Friday. And the first part is that there are a lot of different kinds of gene editing, as Neal alluded to, and a lot of other countries are using the terminology SDN, site-directed nucleases, 1, 2, and 3. So with the 1 being point mutations, the 2 being perhaps when you put in that homologous template and you copy in either a novel portion of a gene or change a bigger portion of a gene, and then site-directed nuclease techniques 3, where you would actually insert either a transgene or a cis gene into the site that is cut by the nuclease. And then there's also others like OMM, oligotype mutagenesis, where it's using a variety of templates and depending on the endogenous kind of nucleases in the cell. So one point that I want to make is that if we mask this kind of diversity of gene editing, we might lose trust from the very beginning. So I've been to a lot of meetings on this where people talk about gene editing as one thing. And so just to make it clear, when you're talking about gene editing, you know, are you talking about the point mutations that largely Neal talked about, or are you talking about inserting transgenes? I mean, it can run the gamut of those. And public confidence is based a lot on, who bears the risk and who bears the benefits, trust, cultural worldviews, history with technologies, familiarity with them, the controllability, whether the people have the right to choose or not choose. And there haven't been a whole lot of public perception studies done on gene editing. In fact, I couldn't find any in the U.S., and I might have missed some. There have been some in Europe where they compare cisgenesis to transgenesis, and generally people are more in favor of cisgenic approaches when you're putting in a gene from the same species into that species. So you're putting an apple gene into an apple gene, or you're using apple sequences and inserting them into apple. But still not as much approval as with traditional breeding for some reason. So people also have a tendency, they're less willing to accept the further genes if the species is further and further away. So for example, a human gene into a plant is less accepted than another plant's gene into a different plant species. So I just wanted to throw that out there. But so I think confidence will be lost if we don't, acknowledge this diversity of gene editing and be specific about which type we're talking about. But yet it's a really big challenge to communicate that diversity. So that's the challenge that we're faced with. Next slide, please. And so here's just a picture of the different kinds of gene editing, and Neal already went over this, so I won't belabor the point, but you have the SDN1 you can see as a point mutation relying on the cell to repair. You have slight modifications where you provide a template, but the modifications are pretty minimal or coming from the same species, and then you have the adding of an entirely new gene. So I think I would just encourage us to define what we're talking about when we talk about oversight or governance and what our views are for these three types of gene editing. Next slide, please. So here's some policy science points. Again, after studying the coordinated framework for a while, we found significant deficiencies, both within the context of gene editing and outside of the context of gene editing. It's a very complex system, and it's very difficult for people, and even small developers of products, even people with some expertise in the system, to figure out which products are covered by which agencies, especially for these newer products that employ gene editing. So the statutes are outdated. There's some pretty strange regulatory hooks. For example, animals are regulated as new animal drugs. Transgenic animals are regulated right now as new animal drugs under the FDA. And so you're looking at the safety and the efficacy of the gene in the animal, and that causes some concern when we think about, for example, the Oxitec mosquito and putting it out into the environment. FDA doesn't have a whole lot of knowledge about environmental risks, doesn't have a ton of ecologists working for it, and so that's just an example of one deficiency that we found. There also seems to be a lack of capacity. These regulatory agencies are underfunded, they're understaffed, they don't get a whole lot of money from Congress, and so there's problems there. And given the volume and the diversity of gene-edited products, I think that's going to be a challenge. And then more on the public side, there's views from people outside of the industry and government agencies that there's a real lack of opportunities for public input. And some decisions are not as transparent as they could be. And some of this leads to a contested policy environment. And so how can we perhaps improve that policy environment? So as you may know, the coordinated framework was taken a look at by OSTP and the federal agencies recently. The White House put out a memo in 2015 to have the agencies clarify routes for existing products under the coordinated framework, but they really didn't tackle gene editing, unfortunately. But it was stated in the long-term strategy document that the federal agencies would tackle that in the future. So next slide, please. So I published this science policy forum that talks about how we had a really kind of opportune moment to kind of re-envision the system, but because of the OSTP and the agencies took more of a clarification route, that opportunity was lost, and that options for policy change weren't on the table. There was little participation and little thought about global harmonization. You can see the map there that the U.S. is one of the only countries in the world that's not a party to the Convention on Biological Diversity, and one of just a handful of countries that's not a party to the biosafety protocol under that convention. So in my view, we missed an opportunity for really figuring out what we're going to do with gene editing. Next slide, please. And I think I talked most about this. Some other things that are going on right now is that the National Academy of Sciences, as you may know, has a study committee on the future of biotechnology and preparedness for risk assessment under the coordinated framework. And in full disclosure, I'm on the committee. I can't talk about its deliberations, but that study is underway. USDA also put out a notice for an environmental impact statement for a new rule on genetically engineered plant products, and under that it has 4 different options that we can talk about in the discussion on Friday if we'd like. I don't want to get into them, but there are various levels of oversight and pre-market oversight for genetic engineering. Again, the distinction between editing and regular transgenic approaches is not as clear in that environmental impact statement as it could be. And then EPA is also looking to revise its guidance or points to consider for genetically engineered algae and microbes. So there's a lot going on right now in addition to the agency's long-term strategy document that came out. Next slide, please. We can skip this. This was the memorandum that came out from the White House in 2015 calling for this process. Next slide, please. Okay. From a more theoretical perspective, just things that I've thought about as a scholar for the past decade or two is that there's always risk and there's always uncertainty, and precaution and promotion are both valid worldviews. And we can get into that as well, but essentially you're either avoiding false negatives or false positives. And I think we're doing ourselves a disservice if we pitch everyone who's somewhat precautious as anti-science, because it's just not true from what I've seen out there. And that doesn't necessarily mean they're anti-technology. It depends on who's using the term precaution and what they mean by it. But it's more of a worldview than one is the right way and one is the wrong way. Second, decisions to prove or not can't be based solely on sound science. And I've used an example of a dose-response curve where you can know the dose-response curve for a risk assessment, but where do you set the safety standard? That's somewhat of a value judgment. So I think we're also doing ourselves a disservice if we don't recognize that values come into play in risk management and regulatory decision-making. And then we also have this issue of product versus process, which we've been kind of stuck on for a while. And that issue, I think, also is if we continue to argue, it's product-based only, that leaves you with 2 options, and that's regulate everything, conventionally bred, gene editing, and transgenic, or regulate nothing. So I just want, if you take that logic, that argument to its final completion logically, that's sort of what you're faced with. And then you're left with the worldview. If you're arguing for regulating nothing, it's a worldview. If you're arguing for regulating everything, it's also a worldview based on values. So a lot of this is based on values. Next slide, please. So, and that was the argument I made in a recent Nature commentary about trying to make these points about science-based and product versus process. And then this is a graph here on the number of products that have gone through USDA through that MI-regulated process. And so that's been increasing, and this includes both gene editing like TALEN-generated plants as well as CRISPR, as well as biolistics, because if you're not using pest sequences to transform, you can put a transgene into a crop and it won't be regulated by USDA right now unless it has plant pest sequences. So we're seeing, a variety of these products, and many of them are coming into the marketplace without any pre-market review, which sort of, next slide please, and this is one of my final slides, which sort of is a hypocritical argument if you think about it. For many years, biotechnologists have used it should be the product, not the process, in order to argue that transgenic products shouldn't go through a regulatory system. But now they're using almost a process-based argument for why gene editing shouldn't go through the regulatory system. So we really, I think, need to be clear about why we're arguing one way or another, and it's usually because of our views and our worldviews about precaution versus promotion and our worldviews about technology. So some of that rhetoric people can see through. And also, if it's nothing new, then why are we funding it, and why is it revolutionary? So if you argue that it's nothing new from a regulatory standpoint, but then turn around and say this is revolutionary from a technology development standpoint, people can see through that as well. They can also see through arguments that we need this to feed the world. Poverty is a huge cause of hunger. Social systems, we have enough calories already. We could feed the world by food waste, reducing food waste, lowering animal consumption, and better distribution. So, however, next slide, please. I think gene editing is going to be really useful, and it's going to be incredibly important to farmers under certain conditions. And it can provide local food security if the crops are directed towards those needs. But my view, after studying the coordinated framework and kind of the policy context for so many years, is that it will be jeopardized unless we kind of start to acknowledge our values when we talk as technological elites, and we acknowledge the role of values in other people's arguments, and don't discount anyone who disagrees with us as anti-science or Luddites or causing us to not address hunger. So that's sort of my view on how we might move forward in a better way. Next slide, please. And this is the last one, I'm pretty sure. We can skip this one. I think I've already talked about most of it. So, I've been talking recently about middle ground approaches moving forward, where we don't just talk, it's all about values or it's all about science, but it's a combination really, and science informs regulation, but decisions are based on both science and values. And so can we find some middle ground approaches for moving forward and kind of addressing this contested climate? Next slide. And that's it for now. And I look forward to the discussion. And again, I just throw these out there for fodder for discussion, not necessarily, to promote this way of thinking.
No, thank you very much. And I think between your two presentations, we really got an excellent context and overview of both the technology options and how the technology works, and the policy societal overlay, which is obviously going to be a big part of our discussion on Friday. So thank you both very much. I'm going to open it up now for any questions. Let me first give an opportunity to Greg and Jim as our co-chairs if there's anything they want to either ask as a follow-up or comment on, and then I'll take others who may wish to ask a question or make a comment. Greg, you want to go first? If you have any questions. Greg, you there?
Yeah, I'm there. Can you hear me?
Yeah, I can hear you. Go ahead.
I don't have any questions at this point. I'll let others others ask them first.
Okay. Jim, anything from you?
Yeah, I'm in the same spot, John. I think these have been great, both from a technical point of view as well as stimulating some thinking around acceptance. So I think it's good to open it up to the group.
Okay, great. So if you have a question, please just jump in. I don't have any written Q&A that's come through on the system, but if you have a question, just identify yourself and please proceed. no questions.
Actually, sorry, it's Michael. Trying to figure out how to get myself off mute. It's Michael Fernandez. Quick
Great,
question,
Michael.
Jennifer. Jennifer, for you, you mentioned the most recent, or the recent coordinated framework update documents, and that, you know, from your perspective, it doesn't really address some of the questions we're talking about here. And I'm wondering if you could just elaborate a little bit more on where that document starts to get towards addressing any of this, and where you think there are still further opportunities.
Well, the case studies, there were two documents. One was a very long, lengthy document about the coordinated framework and which agencies do what. And it came with some case studies.
Yep.
Unfortunately, I think those case studies were not the kind of products that are challenging the framework right now. So, for example, it didn't, the case studies didn't include gene-edited animals or livestock. For example, the hornless cattle that are being developed with talon gene editing. I think it's recombinetics, I think, out in Minnesota, where they've used gene editing to dehorn cattle. So it's much safer for the animal. It's animal welfare. It's safer for people dealing with the animals. But we really don't know how that's going to be regulated, if at all. So FDA, you know, their 2009 guidance talks about recombinant DNA technology and transgenic animals, but we're not sure if gene-edited livestock is going to go under the FDA or not. And I think that's just an example of one of the products that's actually out there right now is sort of knocking on the regulatory door, and that the agencies could have dealt with. Now, I understand that they're under various political and legal constraints, and I don't blame them necessarily. And so I understand there's a lot of tension and things like that. So, but as a scholar, you know, I felt it was important to point out that I think that many of us are disappointed that it didn't really hit these products that are a lot of unknowns and a lot of uncertainty in the regulatory system, and really incorporate them into that year-long study process.
Thanks. It's helpful.
Great. This is John. I've got a question that's come in over the Adobe. Neal, for you, could you just say a word about, Jennifer laid out the SDN123 classification, and the questioner is just wondering if you could say a word about those options and how they relate to your overview of the technology that you provided at the beginning of your presentation.
Yeah. No, thanks. You know, SDN123, kind of technical terms, we try to stay with concepts like delete, edit, you know, replace, which are tracked fairly well along those lines. So, you know, the edit is very much what SDN2 is about. When you cut and don't put a template, so Jennifer mentioned the idea of a template to make the targeted change you're interested in. If you make that double-strand cut and don't introduce a template, you can often get what happens in SDN1, which is a small change that's not a directed change. But at the same time, if you make two cuts, you can actually get a large deletion. And that large deletion would be considered very much like an SDN1 because you have not put in a template to create the change. And then again, the SDN3, it's the idea of inserting a gene. And so that brings you to two different kinds of uses, one of which would be inserting a transgene. And so that's just classical GMO, and in our view, I don't think is relevant to the conversation about, you know, the applications of this technology, the new applications, right? But the idea of moving a gene from one variety to another in its natural location in the same way that you could have done it through breeding is very much, I think, what it makes sense to think about in the context of the discussion we have on Friday. As Jennifer said, there's many different uses, and we should be clear about the ones in a very straightforward way that make sense to consider And those are the kinds of applications that we see under the bucket of breeding. For this group, there's a long history of creating mutations that end up being used in our food crops. This is in some ways a targeted way to do that. And that would come under the SDN1-2 kind of categories. So maybe that's helpful.
Great.
Yeah,
Thank you, Neal. Yeah, go ahead.
And I was just going to add, this is Jennifer, that the EU, at least in their first report of the working group, use the SDN 1, 2, and 3, and other countries are using that framing as well. Although there's a lot of gray area between, as Neal mentioned, between SDN 2 and 3 and even 1 and 2. So it's not the best typology to use. But it is something that other countries are using to kind of make regulatory decisions, from what I understand, in their published reports. And so the EU working group suggested that SDN1 be excluded from the GMO directives, but they weren't sure about SDN2, for example.
Great. I'm going to notice, I know we only have a few minutes left. There was one other additional question from one of our participants about how much, and this could be to either or both of you, how much communication is there going on between those who are working on the application of these technologies to crops, animals, and those working on human health issues? Is there a lot of back and forth interaction in terms of both the science and some of the policy overlay?
I could jump in and just, from a technology standpoint, there's a lot of sharing between all the groups working to use the technology. There are different needs in different settings and different crop or animal or human applications, but very, very common, lots of discussions. We partner with companies that are working broadly in a wide range of applications. And I think at the ethical side, there are some very different issues, and so there have been some separate dialogues, but also some, some very common discussions about the wide range of applications.
Yeah.
Actually, I want to,
no,
Jennifer, let me just.
Sure, go ahead. That's fine. We can just leave it at that.
Yeah, I just want to get this other question in because I think it's an interesting one, and that is, is there a way to detect whether a gene has been modified through natural occurring processes or these technologies that we're discussing at, targeted gene editing post the transition, so to speak?
So I can jump in there, and it will depend upon the change that's made, right? So let's take the waxy example I gave. That particular deletion that we've made, I don't think you would find in anything that historically has been developed, and so it would be relatively easy to show if you had that particular variant compared to other waxy variants that it was one that was made. But in truth, if you have a single base pair change that could have occurred, very easily, those are going to be very hard to detect. There's no question about that. Yeah, so that's the simple answer. I'd say it would depend upon the change made, and in many cases it could be essentially impossible to distinguish between a sort of naturally occurring variant.
Yeah, and I'll just add one more thing to that. So you have to, sometimes you don't have to, but you can, some people put in the editing machinery, and those are, trans genes, and then what they do is back cross them out. So, and those are null segregants, I think they call them, and so that, just like Neal said, then you're left with, if you take the trans genes, you put them in to do the editing, and then you take them out, then you're left with just the change, and then that, it'll depend whether it's found in nature or not, whether or not you can detect it. So I just wanted to add that point about the null segregants.
No, very helpful. Thank you both, and I think we will wrap up here. Let me just say we got another question that Jennifer, I'll just let you know it, which is the questioner asked about your views on why the U.S. is not acceded to the Biodiversity Convention. So perhaps you can think about that one, and we'll put that on the Friday agenda.
Okay.
And there may be some other questions that we just didn't get time to cover today. I apologize, but this has been, I think, exactly what we were hoping for, which was a really solid overview of the issues and a chance to get some clarification and additional information. If you have other questions, we are going to spend some time at the beginning on Friday to kind of wrap up any additional information exchange about the technology. So if this has spurred some additional thinking on your part, we will have a short period at the beginning on Friday to have Jennifer and Neal and others respond to any additional questions. So please keep those in mind. And this webinar will be on the website this afternoon. So if you want to review any part of it, or if you're talking to other folks you know are coming on Friday who say, oh, heck, I missed that, please let them know. And we will as well, that this will be accessible between now and Friday. Deb, let me just turn it over to you to see if you want to say anything before we close.
I just want to thank everyone for their participation, and particularly to our speakers. Thank you, Neal and Jennifer, and I look forward to meeting you in person later this week. So with that, let's sign off and talk to you all soon.
Thank you all very much.
Bye.
Thank you.
Bye-bye.